Welcome back to The Explainer. Today, we're taking a forensic deep dive into one of the most complex mysteries hidden right inside our DNA. We are going to explore exactly how we discover structural variants. Now, these are massive, messy genomic changes that totally rewrite the biological script. Think of us as genomic detectives today. We're going to examine the biological incidents, analyze the digital clues left behind by DNA sequencing, and finally, we'll meet the algorithmic investigators that were built to crack the toughest cases. Here is a quick look at our roadmap for this explainer. We're going to define the SV challenge, decode those alignment signals, profile Lumpy and Manta, assemble the GATK task force, and then, finally, look at future horizons. Let's jump right in. All right, part 1, the SV challenge. Let's define the incidents we're actually looking for. For a really long time, genomic research was kind of hyper-focused on SNPs, single nucleotide polymorphisms. And, that makes sense. SNPs are just small, single base pair changes. They're essentially just tiny typos in the genetic code, which makes them relatively straightforward to detect. But structural variants, SVs, they are a completely different beast. By definition, we're talking about genomic changes larger than 50 base pairs. So these aren't just typos anymore. They are massive structural overhauls. We're talking deletions, insertions, duplications, and inversions. And because of their sheer size, SVs have a profound impact on gene dosage, function, and regulation. They're actually directly linked to developmental disorders and cancer. But here's the catch. Detecting them is incredibly difficult because we still mostly use short-read sequencing technologies, which give us reads of only like 100 to 300 base pairs. Trying to find a massive structural variant across 3 billion base pairs using short reads, it's honestly like trying to comprehend a massive sprawling novel by only looking at scattered fragments of sentences. Now, if you look at how we represent these basic SV types in a standard VCF, or variant call format, it's super helpful to think of a deletion like missing pages that have been ripped right out of our reference book. And an insertion, that's like a brand new paragraph pasted randomly into the middle of a page where it literally just doesn't belong. Because the DNA itself is structurally altered. When we chop up a patient's genome into tiny pieces to read it, those pieces simply won't match up perfectly against the standard human reference genome. Moving right along to Section 2, decoding the alignment signals. This is where we start finding our digital clues. Okay, so our first major clue is the read pair signal. When we sequence DNA, we usually read both ends of a short fragment. Naturally, we expect those two ends to map to our reference genome at a specific distance from each other, facing inward. But what if they map way too far apart, or way too close together? Or what if they're pointing in the wrong direction entirely? Well, that gives us a discordant read pair, and that is a massive red flag that a structural rearrangement has occurred right between those two ends. Next up is the split read signal, which is really cool. This happens when a single continuous short read happens to perfectly span the exact junction of the mutation, which we call the breakpoint. When we try to align the specific read back to the reference genome, half of the read aligns to one location and the other half aligns somewhere completely different. It is exactly like finding a torn sentence where the first half matches page 10 perfectly, but the second half perfectly matches page 45. It gives us incredibly precise base pair resolution of exactly where that break occurred. And finally, we've got read depth. Since sequencing is generally pretty random across the whole genome, we normally expect a relatively even coverage of reads everywhere. But if we suddenly see a massive drop in coverage in one specific spot, it strongly implies that piece of DNA was deleted in our sample. On the flip side, if coverage suddenly spikes, it implies a duplication. So read depth is kind of like weighing our document. If it's suddenly way lighter or heavier than we expect, we know entire chapters have either been deleted or duplicated. Which brings us to Section 3. Let's meet our first digital detective, Lumpy, in its probabilistic framework. Historically, older algorithms would basically just use one of those clues we just talked about to find an SV, and maybe, if you were lucky, use a second clue later just to verify it, but Lumpy completely changed the game. Lumpy's core strategy is a probabilistic framework that naturally integrates read pair, split read, and read depth signals all simultaneously. That's the key, simultaneously. Instead of just guessing a single exact breakpoint coordinate right out of the gate, Lumpy uses math to create a probability distribution that spans the whole predicted region. It essentially takes the uncertainty of our clues and maps them to probability intervals, combining multiple weak signals into one really strong confident call. And you can see the impact right here. Lumpy has a massive advantage in sensitivity, even at very low sequencing coverage, like we're talking just 5x coverage. Because it combines multiple signals into a joint probability distribution, it pulls the real signal out of the noise far better than older tools that process those clues sequentially. So when evidence is scarce, which honestly happens all the time in low-coverage data, or in really heterogeneous cancer tumors with low allele frequencies, Lumpy yields vastly improved sensitivity while still maintaining a highly competitive false discovery rate. Just take a look at this diagram. When we compare deletion discovery across different callers on a high-coverage sample, Lumpy is discovering hundreds of true, unique variants that completely slip past other detection algorithms like DELI or GSV Pro. By not forcing a single-point prediction too early and instead letting the probabilities naturally guide the clustering of the evidence, Lumpy uncovers genomic secrets that other algorithms literally just ignore. All right, Section 4. Let's talk about MANTA. This one is all about speed and assembly. So while Lumpy uses probabilities to build its case, MANTA takes a totally different route. It relies on a graph-based methodology. MANTA's core strategy involves two major phases. First, it scans the genome's paired-end and split-read evidence to build this massive graph of potential break-end associations. It maps out all the places where distant parts of the genome seem to be incorrectly connected. Then MANTA takes those clues and literally tries to reassemble the torn pieces at the scene. It performs local sequence assembly to resolve these breakpoints down to the exact base pair. It's an incredibly unified workflow that handles both large structural variants and those tricky medium-sized indels, and it is highly, highly optimized for comparing somatic tumors directly against normal tissue samples. To really put the computational engineering here into perspective, MANTA can process a massive 50x coverage whole human genome in less than 20 minutes on a standard 20-core server. Less than 20 minutes. That level of speed is absolutely staggering. Combined with its accuracy, this efficiency makes it an absolute powerhouse for large-scale sequencing pipelines. So let's compare our digital detectives directly. As you can see, MANTA absolutely dominates in speed and somatic tumor analysis, while Lumpy matches it with incredibly high sensitivity in difficult, low-coverage scenarios, thanks to its probabilistic modeling. You might be wondering, well, which tool is better? The truth is, because they use completely different algorithmic approaches, assembly graphs for MANTA versus probability distributions for Lumpy, they actually often catch entirely different subsets of true variants. All right, moving on to Section 5, GATK, Single Sample Workflows. Let's assemble the ultimate task force. GATSV really does act as this ultimate task force. Rather than just crossing our fingers and relying on a single tool, this pipeline actually runs MANTA, WAM, and GATKG-CNV as its raw calling algorithms. It takes all of those varied outputs and completely integrates them. But the real magic of this specific single sample workflow is step 2. It compares your single patient's data against a massive pre-computed reference panel of 156 high-coverage genomes from the 1000 Genomes Project. It uses machine learning models trained on that exact cohort to filter out sequencing artifacts, refine the exact variant coordinates, and deeply annotate the final structural variants. It basically brings the true power of massive population studies right down to your single clinical sample. Now, just a really quick heads up for any practitioners actually looking to implement this workflow locally. If you're used to looking at large public databases like Nomad SV, you'll know they use a tool called MELT to detect mobile element insertions. However, because of licensing restrictions, the publicly available Gate shingle sample pipeline actually skips the MELT tool entirely. You definitely want to keep this in mind because your output is naturally going to be slightly less sensitive to mobile element insertions compared to those large published joint call sets. And finally, Section 6, Benchmarking and Future Horizons. Practically speaking, what you can actually expect to find depends entirely on how deep you sequence your samples. If you sequence lightly, around 10x, well, tools like Lumpy can still find large structural variants over a kilobase for sure, but smaller, complex events are going to be practically invisible. At the typical clinical standard of 30x, you'll comfortably detect most SVs. But if you really push to 50x coverage and beyond, you provide these algorithms with an absolute abundance of split-read and read-pair clues, which allows them to resolve incredibly small SVs with practically flawless breakpoint precision. I want to leave you with this final thought. We've just explored some genuinely incredible feats of software engineering here. These tools are literally jumping through massive algorithmic hoops to piece together tiny short reads into massive structural variants. But as new long-read sequencing technologies, we're talking reads that are tens of thousands of bases long, become cheaper and much more widespread, how will our trusty digital detectives adapt? Will tools like Lumpy and Manta evolve to process those long reads? Or are we looking at an entirely new generation of algorithms just waiting to solve the genomic mysteries of the future? It is absolutely something to think about. Thanks so much for exploring the science with me today, and as always, keep questioning the data.